Okay. Okay, very good. Good afternoon, everyone. Welcome to the next piece, our January edition of the CellCan webinar series. Thank you very much for joining us. It's our great pleasure to be able to host Dr. Michel Trombley and the McGill Regenerative Medicine Network. I'm quite pleased to say this is something that is near and dear to my heart. A few moons ago, Michel and I sat down and we had ideas around what a network at McGill might look like for cell and gene therapies and how to get that kicked off. So very pleasing to see that this is a bit of a coming of age opportunity for us to talk a little bit further about the network, its progress, and where things are headed. Before we get started, I'll tell you a few words about CellCan. We are a federally funded Networks of Center of Excellence program that is dedicated around focusing on the niche of cell and gene therapy manufacturing, with our vision and mission to optimize manufacturing practices by creating a common seal of quality to increase the capacity and help rapidly translate therapeutic products into clinical production. We have 4 main business priorities that we centralize our business around. Hopefully many of you have seen this already. Building a strong Canadian network, advance regulatory standards. We do this by working closely with the regulators in Canada, Health Canada, and even Environment Canada and Climate Control. Generate effective outreach. This webinar series is very much a strong part of that program and helping to position Canada as a world leader in the sector. We're extremely pleased to be able to announce that we've launched our online GMP Training Institute. We've started off with 2 learning streams and learning paths, as we like to call them. The first is more of an executive snapshot of what GMP is all about in under 60 minutes. It's very much a global overview with covering the essentials of what manufacturing under GMP is involved in our sector. And as well, a more robust curriculum around our more crowning achievement, the hands-on Gmp Workshop, which we had hosted annually at the CTC in Montreal at Maisonneuve-Rosemont Hospital. We've taken to a more robust, interactive online learning format. It's designed to take in about 15 to 20 hours in total time, including all of the interactive modules and assessments. And we hope very much that you'll take a closer look at this and understand what it's all about. We put all of the curriculum on a learning management system, which is very easy to navigate, it's easy to use, and it facilitates a learning stream. So any HQP that takes any of the modules on learning programs will have a record of training throughout. It gives us very interactive computer-assisted learning-based approach to the modules. And of course, there are assessments throughout to make sure that the knowledge is taken up to the highest level. We've created some of our own content that's more specific to our sector, cell and gene therapies, but as well, we've engaged with a partner that produces professional content within all fully narrated and professionally developed modules, such as this one on bioreactor-based production of cell culture. We're very pleased to say that one of the superclusters within Canada, the Next Generation Manufacturing, or ENGINE as it's better known, has supported our program. They've reviewed and validated the curriculum, and they now offer it as part of their AMP Up program, which is a skills, an upskilling part of the overall program at ENGINE. This applies to the GMP Virtual Hands-On Workshop, and ENGINE's willing to cover up to half of the costs for employees of ENGINE member companies. The good news, it's free to join ENGINE, and full details can be found at engine.ca/upskilling. Please don't hesitate to take a look at that website. If you didn't get the information, reach out, contact us. We'd be very pleased to talk further about that with you. So without any further ado, and the point of turn it over to our expert today, Michel Trombley. He's a full professor in the Department of Biochemistry and Oncology, as well as the James McGill Professor and holder of the Jean and Jean-Louis Lévesque Chair in Cancer Research at McGill University. He was also the former director of the Goodman Cancer Center at McGill. So many of you are already familiar with Michel. His reputation precedes all of this, and Michel, we look very forward to hearing your talk today. Thank you.
To start sharing, thank you, Craig. Indeed. So, do you hear me well?
Very well.
Yeah, that's fine. Okay, so the picture of Corinne is there, Corinne Osley, and she, because of a family health issue, she was not able to come today. So she really sorry about that. I'll try to give her justice to all the bioengineer at McGill. I will do my best, but she's the one that you need to talk to for all these robotics and more. So the talk today will be, let me see if I can change this. How do I do this? Here. Okay, let's see. So there's three sections of the talk. Mainly, I'll tell you, as Craig mentioned, and he was there indeed when we start talking before we made the network. And Craig was an important person when we gathered together and start thinking about this, and that led to the writing of a strategic plan for McGill University in stem cell and regenerative medicine, and this is the MRM is the result of that. So what I'll tell you is a bit an overview of the MRM. I'll talk a bit about bioengineering facilities and expertise, but I'll come back to that a bit at the end. And I'll give you a case study where several groups at McGill and companies have put together a project in dendritic cell-based cancer immunotherapy. So that would be the presentation. I'm going to look at my watch to make sure I'm not passing the time, and Craig or Vanessa can certainly tell me. And thank you, Craig and Vanessa, for the invitation. I think I'm on a timeline that would be scary, but we'll try to keep this. I'm not sure I can get it out of that. So McGill, in fact, has a long history on stem cells. This gentleman here, called Charles-Philippe Leblanc, during the Second World War, immediately after, came to McGill and became the chair of the Department of Anatomy and Cell Biology. And he's the one who published in the first paper the term stem cells for animals and the idea of stem cell renewal theory. So McGill has a long history, yet when you think about this, not much was happening at McGill when I was there, and even Craig was there. You know, even the Faculty of Medicine did not realize there was a lot of people working on stem cells at McGill. So, right directions. So McGill, in the past few years, has established several transversal large program. I'm sorry about that. And including, for example, the Institute of Cancer, the McGill MI4, which is immunology and infectious disease, and many others. The MRM is one of them that go across the entire McGill community and reach to people doing stem cell research and regenerative medicine. We were approved in December 2018. We were given a small budget that we're working hard to expand, but this is the insight that after Craig left, that's what happens. So we have now the MRM. Let me tell you a bit more what this looks like. So if you want to have know more about this, you just go to MRM McGill in your search, MRM McGill, and you'll find the site, and you'll have all the information that I will present you today. Our contact is Marine Christin. Marine is a fantastic, she was a postdoc at McGill working in part in stem cells too, in the neuronal field, and she joined us as the coordinator, and we're very lucky. So we had five aspects of our mission, if you want, when we get this strategic plan. We want to identify McGill's strength. We did not know who was there, and we in fact published. McGill is one of the top 4 universities, sometimes third, sometimes fourth, publishing in the field of stem cells and regenerative medicine. So it's very surprising that we did not know who was publishing at McGill, and this was the first mission of it, and try to bring together who's working, the field that we're working, get us going, and then also partner with other people like the CCRM, the CellCan, and many other ones, T-Cell in Quebec, for example. So I'll show you a bit more of this. I'm playing with this now a bit too much, but okay, let me see. That works. So MRM now, if I give you in a nutshell, there's 100 different PIs working on stem cell and regenerative medicine that are members of the MRM. And there's 500 people, such as student, postdoc, staff, and so there's an immense. I find it immense from the 4 people who started the network initially. There's a large group of people now recognized at McGill and doing work in those fields. There's about 25 departments are touched by this. 5 faculties. You can see medicine, science, engineering, and all the research institute of the McGill affiliated hospital have people, and the institute recognized the MRM as a network for McGill University. So quite a lot of we succeed in this first part of our missions to really identify who is there and make a bit of sense about who we are. And it's easier after that to come to positions like this webinar and other partner to describe what we do and be able to share. So we are also, since we've done this network, we have worked with many people in Canada. Not the network is not always an associate member of these different organizations, but we have people in all of the organization present or collaborating with them. We have also sent people to Till and Mekelo meetings. I think the past 2 years we sponsored probably 40, 45. I'm sure Marine is on the line and she will correct me later on, but it's something like that we have sponsored. Also, we were 2 years ago at the International Stem Cells and Gene Therapy meeting in Montreal, part of the big, very nice booth that was organized. I think it was you, Greg, and Vanessa that worked on that initially, and this was a fantastic event. And we have organized also meeting. Maxime Bouchard has organized a meeting on stem cells with many people from the European community and outside and in Canada. So all of this, that having at least a tour of who we are and what we do, it has been possible to fundraise money and be able to sponsor people and organize and do things as a community at McGill University. The second one and the third one you'll see afterward is really promoting basic research and in stem cells and regenerative medicine. So all the people that are in the network, the only criteria we ask is that they do a bit of this research. So they have to be able to do stem cells and other type of regenerative medicine work, and they're happy to join. We don't ask them to pay. We just ask them to be a good citizen to the community and enjoy their time. So I'll show you a few things we do. There's people doing, and Matt Kinsella, for example, in bioengineering, do a lot of nanotechnology. Corinne, of course, in the stem cell bioprocessing lab. There's a remarkable one at McGill. They invested quite a lot of money in engineering to do this. I'll show you a picture later on. IPS cells core facilities with Carl Ernst and Thomas Durkin at the MNI that are doing really nice core, open again to the entire community of scientists at McGill and beyond. We have, in addition to these group of people, we have done something I feel interesting and useful for McGill University, is the fact we made partnership with all the core facilities that were interested to work with the MRM, and the fact that our members were working with these cores. So proteomics, genomics, and many other ones in stem cells, iPSC, etc., etc. So what we've been doing is really to make small grants, and these small grants can go as much as $8,000 to fund the use of this core by our members. So we make calls. Our members, if they need, for example, to have an expensive experiment on single cell sequencing, so they can apply to the MRM to get this small grant, and they can use it for our core facility. And as you may know, all of you in your own institution, it's very hard to pay for core facility at university. So in a way, it's helping our constituent, and at the same time helping our core facility to receive work and strengthen their revenues and making sure the core are up to date. Another one that it's not a core that we are negotiating with them now with Korean is the one that is present in the engineering facility, engineering faculty. And there's many of these bioprinting, 3D printing. They have a large segment of their faculty that are doing this. You can see, for example, Korean pointed to me. Again, this is a Korean space, so hopefully I'm not destroying the reputations of the bioprinting facility, but this 6-axis robotic arms allows you to make organs. You can make intrusions of cells into a 3D segment that you can generate. So there's quite a lot of work that's done there. Several papers have been published and more coming. So they are a very active group on bioprinting from hearts to other organs or tissue, blood vessels, etc., etc. So if you're interested, please contact Corinne. She would be happy to tell you more. So the third one is the clinical pipelines. And especially at the CellCan, it's difficult to develop. Not everything would be the same. We're not necessarily doing all transgenic modifications of iPSC to put in a patient, but they are, and the example I'll give you later is not such like that, but there's many things that can be done. One group that works very well, and again, Craig knows that group very well. He was there. It's a lab that was developed by Stephen Paraxivas at McGill University. It's at the Genome Center, and it's a cell transfer lab. You can grow, and it's Health Canada certified. And mainly Stephen is a diabetic person working on diabetes. He's not diabetic. He's working on diabetes, and he's doing islet transplant into type 1 patients that are becoming. They could not have any more insulin injections. So this has done, several patients have been done there with success. And so this is one core facility which is really remarkable at McGill. It's now used also by the people at the MNI for other types of clinical trial. Lady Davis Institute had a clean room in the past. They have restarted doing things there too. And we hope that with Nicoletta Eliopoulos, there'll be more of the use of this clean room for certain types of work, bone marrow transfer and other things. Pierre-Laneuville has a small lab that very nicely done though at the MUHC Research Institute. They call the cell transfer lab. So it's not a lab that people do genetic manipulations of cells, but it's used for cord blood transfer. It's used for the work I will show you on dendritic cells that, as you can see on top here. Oh, but you don't see anymore here. Vaccination with prime autologous dendritic cells. I will show you a bit of that. The work of Jean Chervenkov also and mesenchymal stem cells are also using. So there's about 12 clinical trials in line for this small lab at the MUHC, but a very important one. It's run by Pierre and Linda Pelletier, particularly. I should mention that. Linda was also a very important person when we started the network at McGill University. And without her, I think we would never started, I should say. So finally, at the end, I just want to remind you that the Schreiner Hospital are doing quite a lot of limb reconstructions. They work on stem cells for bone, for also cartilage, and Hamdi and Al-Ajad are some of these people that use also 3D printer to make bone and reconstructions at the Schreiner Hospital. So this is a variety of things, and I think the clinical applications are increasing. The partnership between McGill and other hospitals and centers that they can do cell transfer also is increasing very remarkably. So I'm just going to mention, you mentioned a nice training program, Craig, which I think is really cool and very important for manufacturing. So we have not that, but we have something for our students, and hopefully they'll be the future people taking your training. And that's the idea here. We made a graduate certificate in regenerative medicine at McGill through the Department of Experimental Medicine. So what it means when you graduate with your PhD and with this certificate, you have that stamp on your diploma. We did this really rapidly, and it was very nice. Terry Hebert and Linda again worked on that quite a lot. We have about 40 courses that were existing at McGill on stem cells and development biology and application in the clinic. We created 3 courses on clinical application of regenerative medicine, directed reading, and also a stem cell bioprocess that Corinne has developed. So with that, we think our students, there's several people now, it's increasing the number, they are interested by experimental medicine and this program in stem cell and regenerative medicine. And we hope they will, when they graduate, they'll be the people going around in Canada and taking other positions or staying at McGill or coming back to McGill with that. This program also is a place we advertise for graduate positions for our members. We give travel awards to students. So all what related to training and graduate student program are done at the MRM through this program to trainees. Finally, you may know Bartna Nopers. She's been working fantastically for many, many years at many places in ethical issues of genomics and development, embryo and stem cells, of course. And so she's part of her center is part of that. And we have several people at McGill in ethics, in law, and other people interested in social implications of regenerative medicine. That's part of our university's role and other university in Canada also to show the good and the potential difficulties of these technology. And I think that's also the role of the MRM to present that to the public and our colleagues. So that's the last thing. I think I have that. So we have on our website, we create seminars, we have conferences, we have delegations meeting people at McGill. We have a very nice little MRM Insights. It's more opinion pieces on stem cells and regenerative medicine, ethics of from the members of the MRM. So it's fun. Some of them are very serious when we talked about regenerative medicine and COVID. And other ones are more opinion on genomics, and so it's interesting, and we give pretty much a bit of freedom for our professors to give their thoughts and not their papers. So that's also fun to read. Okay, so I'm not sure how far I've been pretty fast on that, but I think it's on time. Unfortunately, I had
You're doing well for time.
Is that okay? So I'll try to slow down on some of the thing coming. So the case study that I want to show you is an example of a different group that came together to develop an immunotherapy process that I think because the simplicity of this immunotherapy, it's easier to develop for a cancer vaccine. So that involves, of course, the MUHC Research Institute and the lab that Pierre and Linda are doing. It's involving the stem cell lab of Corinne Osley, Saint-Gobain, which is a huge company, but they have a very important section of that company on blood vessels, tubing for blood and bags of blood, and they are interested, of course, to develop tools to increase the growth of cells and improve and optimize the productions of cell for cell therapeutics. So and the little one at the end, in the middle here, Canet Pharma, is a company that I started a few years ago. I have to declare that here too, but it's a company that has moved quite a lot in different directions, and especially on this dendritic cell technology that I will give you this case study, and we'll be talking about growth, clinical trials and perhaps simplifying the logistics of stem cell therapy. And that's a call for Craig, to look at this and see if it's good. So the technology here is very simple. Also, it comes from the fact that two tyrosine phosphatase, TCPDP, PTP1B, or PTPN1 and N2, these enzymes are negative regulator of cytokines. And several types of cytokines. So if you take an inhibitor of these enzymes, you can reactivate many different stem cells and also immune cells. So that's what this company has done, Canin Pharma. There's a series of compounds that were made, and they're extremely specific to these two enzymes for different reasons that I don't have to go into, but the structure of these two are a bit different than the rest of the 105 tyrosine phosphatase that make these compounds quite interesting, stable in water, and very effective. So the company, as they are going is going in many different directions. The fact that we can potentially inject these compounds in people to treat cancer and immunotherapy. We also develop, and this is in partnership with Jean-Sébastien Delisle at Maisonneuve-Rosemont, this compound K8484 it was able to reactivate not activate, but protect CAR T from exhaustions and few other things. So we see it as a T cell enhancer, but this is all ex vivo. And the dendritic cells, it's the technology I'm going to present to you today. So you can activate very well dendritic cells to present antigen to T cells to kill tumors by using a compound, a simple compound that is relatively cheap to make and be able to make a GMP relatively easy. So this is what I'm going to tell you now today. So we got a lot of flack. I say we because I'm part of this company. The company had because we decided to go in chronic myeloid leukemia, and for an oncologist, this is kind of a very not serious cancer. But as a proof of concept, I will try to convince you that this was extremely useful for us for different reasons. So CML is the cause of CML is the translocations of two chromosomes, 9 and 22. And what happens is the BCR gene, part of the BCR gene, is become associated with the kinase domain of ABL. And that makes a new protein, the BCR-ABL protein. That is, when the protein is made, the BCR side of the protein can dimerize, and then you have a dimer of these hybrid protein that activate the kinase, highly activate the kinase, and makes this tyrosine kinase a true oncogene. Then that will cause, once you have these translocations and recombination, a chronic phase where you see cells of the myeloid compartment that will expand, and this is often how clinicians will be recognizing the amount of cells that are there, and they're pretty much asymptomatics, but if there are too many of these cells, then there's some issues. That period, if it's not treated, that period, the chronic phase, may accelerate with other mutations, and you can have an accelerated phase with these new mutations that then will show to patient some phenotype or symptoms, increased tiredness, weight loss, and large spleen. So that's very clearly diagnosed. And finally, other mutation can still accumulate with this growth to give by the fact there's an increased genomic instability, and then you can have a blast crisis and development of acute myeloid leukemia, AML. So the advantage of targeting this for our technology, well, first of all, I should say that there's an increasing number of these patients for different reasons. One is that there's outstanding kinase inhibitor to treat the patients. And even if you diagnose with CML and you follow the larger populations of these CML patients, you can see that the life expectancy is getting closer and closer to normal. So clearly stating that the therapeutics right now are working very well for CML. And however, so these patients accumulate. They're not dying in 5-6 years. They do live well, and we'll talk about some issue. And they with that they will have to take these drugs for the rest of their life. So that's there's few issues. These are the type of compound that people are using. Imatinib was the first one, very well known compound for leukemia treatment. But there's a slurry of other compounds that develop because of competition, other company wanted, but also because people became resistant to the drug, and the tumor became resistant to the drug, and then other therapeutics had to be used, and also there were an increased amount of side effects for some of these drugs. And each time the new generations of these compound codes showed up, since the population is lower and lower and slower and slower, I mean, the population for treatment, then the cost obviously went up. So the cost of treatment is over $100,000 for some of these per year for some of these compounds. So you see on the left side here the life expectancy, that this is the normal populations, men and women, and what you see is that they reach very close, and almost it doesn't, it doesn't go totally, because of this blast crisis that can occur. But these drug functions. So what I tell you is that in CML, the TKI are not optimal for different reasons. First, the side effects. There are several that gives a very severe side effects: liver problems, edema, heart failure. So these are serious effects that it can occur. Treatment resistance and cancer progressions, and the high cost, as I mentioned. So this is an interesting target already, but there's more to this. Pierre Lanneville is an immuno-onco that treats the people in CML and other leukemia and lymphoma, and produced a little paper not long time ago where what he shows is a series of clinical trials on CML. And what those clinical trials, in a nutshell, they were asking the patient to stop taking the tumor, the kinase inhibitor. And they were able to do this because the way that these patients are monitored, just a blood test is done every 3 months or a different period of time. And you try to see where the BCR-ABL genes is present or not. So it's a great way to monitor the presence of the tumor cells. Some patients will live with the TKI with a relatively low level of tumor cells that you can monitor, and they're varying for patient to patients from time to time, or they're very stable in some cases. And in other populations of patients where you cannot detect the tumor cells anymore. Most of these clinical trials, when these occur, they try to see, can we remove the TKI treatment and see if the patients will be cured. And indeed, about 50% of these patients, when the patients that do not have detections of tumor cells in their blood, if they stop taking the TKI, the kinase inhibitor, about half of them they will be tumor-free. They will be the treatment-free remission, so the tumor cells not come back. The other half, the tumor cells come back very rapidly. So it's interesting because it shows that you can potentially, at least for 50% of them, having remove the treatment and have a pretty good life if you can follow them up in case it comes back. But the other half, potentially, if you have another treatment than TKI, you may be able to reduce the number of these patients to. I'm sorry, I'm checking my time to make sure that we reach the end. So the idea for us was to use our dendritic cell technology into a phase 1 trial, and this has been submitted to Health Canada. We have very good review, few things to do that we're resubmitting now. And the fact that we're taking it's patients that are taking tyrosine kinase inhibitor, they could stop and they can still keep treatment. So we can almost do combination therapy. We can use our dendritic cell treatment for some steps in their disease, and then hopefully we can show, and we'd like to have at least one and more log reductions and potentially the disappearance of tumor cells in the patient that will be selected. And it's low cost because many of the tests are already paid. And it's, as I said, the drug is relatively easy to do, and the logistic also. So the treatment, I will pass that very quickly, but it takes about 8 days to take monocytes from the patient, differentiate them, put the compound with the tumor antigen. BCR-ABL are very well known peptide for immunotherapy by dendritic cells, and then you isolate these cells and fully functional. You wash the compound away, and you can put the cells with no compound activated, ready to go for the patient. So that's the idea. So if you look at the patient per se, the patients will come, the blood will be taken. Interestingly enough, we don't need to have leukapheresis. Only 4 tubes of blood is enough to have enough cells to do all of this for 3 to 4 treatment. So this is the plan that we propose. We examine this in the preclinical testing, and so we are ready to do. So the treatment, you take the monocyte, you do the 8 days that it takes to do that, and you can return to the patient the cells. So there's no genetic change of those cells. The compounds have no side effect. Everything was tested in preclinical work, and the cells look perfectly normal, and they return. They do their job for a few days, and since there's no compound, eventually the phosphatase comes back and will do its job, but already the dendritic cells would have prime T cells for killing the tumor cells. So this is a simple procedure for dendritic cells that we think can fit extremely well as a proof of concept with CML. So that's the last slide of this story. I will pass on something else, but just to show you, we hope it offers a real cure for chronic myeloid leukemia. There's no side effect of dendritic cells. It's not like CAR T. The only thing is there's a bit of rash when you transfer cells into patients, and they'll be able to stop the tyrosine kinase inhibitor. And obviously the cost of this is very cheap because it's all autologous transfer, and there's a very short time, no population of cells they have to expand. So we can, if people have questions, they can ask me at the end. So what I want to tell you about is really another thing, which is eventually will be in phase 2. We'll be working that with perhaps CCRM or CellCan, I don't know. But in the meantime, we want to see one of the big issues on dendritic cell therapy. Another one is the logistic, which is very difficult. And again, Health Canada will certainly push for a central facility until other things are developed. But we think that this model of we're trying to do may be used on every single hospital, because it's not, the cells are not genetically modified. The compound are just treated. It's seen as an ancillary compound by Health Canada, so that it's not seen at all as a genetic treatment or a severe treatment of these cells. So nothing else than treating with growth factor as people get in many blood transfusion. So we did several things to the logistics of that. One is to develop a therapeutic kit. So each patient will have this same box here, and I made a little cartoon. You can see I made it, how bad the cartoon is. The point is you have all the things that you need, interferon gamma and PLA. That's the concoctions we have to do with the different tumor antigen. You change patient, you change tumors, you can change also the tumor antigen. You can put more than one. So you reducing the chance of resistance, etc., etc. So there's advantage, and these kits can be given to hospital for the entire durations of the treatment, and it's stable for that. So that's the idea of the logistic. But there's other things we've been doing, and this is concerning also Saint-Gobain and other ones. We are working through the Oncopole with Claude Perrault and other ones to use this technology and to see if we can develop it for AML with different types of peptides. However, and this is a project that is going on the side. An important one, though. So what I'm showing you is a little scan, a little cartoon that we put on the web when we got a very nice grant. Corinne Osley was the main PI of this, with the partner of Saint-Gobain, the company that does these bags and tubing for hospital. We were brought in by Canier Pharma to bring our compounds and our procedures, and again the lab of Pierre to try to get all the cells to test this. What's the idea here? The idea is to develop eventually, so first of all, improve the matrix of bags where you grow cells, grow stem cells. In this case, we focusing on dendritic cells, and also try to have a bag which will have, this is a depiction of it, but we would like to have eventually a bag that will have multiple spectra where we can have a closed system from beginning to the end. The only thing you will need is you will need a hood and a tissue culture, an incubator and a tissue culture hood. That's the only thing you will need for this, and that's what we think we can do. So I'll show you a bit of that, so we can take the monocyte in one side, have different time in the 8 days that you can isolate the cells in the sealed bag, and then be able at the end to collect the dendritic cells, activate it, and give it to the patients in a single round without any large increase of the cells. You don't need it in this particular technology. So this is I want to present now work that was done with the team at Saint-Gobain and Corinne, and give you an idea that the things they're doing to improve the system. And that will be the last part of the presentation. So they tested bags that were generated at Saint-Gobain and other places, and different matrices, fluoropolymer bag, for example, versus tissue culture polystyrene. And the idea here is that there's a lot of advantage to have a bag over this, the safety, the sterility of it. And we think that that can be done in a much nicer way and improve significantly. Corinne's group has done several things. One is to look at how cells in bags attach or in plastic attach. So there's three parts of it that you can follow. First, cells in suspensions will have contact to the matrices, and so they can have different cell adhesion molecule that will contact and detach, and the cells have a certain time that they will be attaching to these matrices. That's important also for how the cells will grow and they're expanded. Adhesion is another thing. If the cells need adhesions, could they attach? Could they then contact other cells which are beside them? And finally, when they're very solid adhesions, then these cells are good to produce factors, to differentiate and do other things. So they follow this with the different matrices. This is an example of the height we're talking about, the height sensors. And what you see here is that fluoropolymer, and I'm trying now to remember what I was told. I came back to the desk and being told what to say, but I'll try my best again. Take that with a grain of salt. But my understanding with Corinne is that, for example, fluoropolymer will attach much faster than polystyrene. So there may be some small advantage as you can test matrices like that. But if you do this at a longer term, if you do this at the at a higher level of cells, then these advantages will accumulate. Another directions that they did is they try to see how cells attach to these matrices. So, Craig, you talked about your incubator, expanding the cells. That's true. And eventually, though, they do put protein, they secrete, and these protein will attach even if you have, changing in the media. So, what is the matrices that holds those cells to improve their growth and their expansion? So what the group has found is that, interestingly enough, it's not just one layer of protein. There's several layers, at least 2 layers of protein. And these you can then change the type of protein that you have, and you can, again, force one way or the other the differentiations and the expansion of the cells by choosing the right matrices. So it's not just a type of vessel that you just put stuff in. I think what the cells will feel and what you can do to modify this is very important and gives in the future the directions of where I think cell therapy will be using, various techniques they will be using to improve this. And other directions that, again, this group of people have done, and to give you more idea, is so Yeah, it's nice to have bag, it's nice to have flask, but if you need to test 20 different compounds, or if you need to test many different protocol, it becomes very hard, especially if you have to look at markers, and these antibodies are very expensive. So what they developed in with Saint-Gobain, the group of Korean, is a smaller and smaller bag, and you can see some of them have multiple compartment, but they were able to develop 2 ml bags and then test these bags to see are they producing the same thing than others, and that's what you see here, different vessels. And we are still in the process of that, but these bags seems to work very well, and there's not very significant changes that you have a large bag of 750 ml or a small bag of 2 ml. That would be enough now to develop the technology to go forward. I'm almost there. I have a few more slides and we'll be done. So again, another approach that you can see that all these components of developing the matrices to develop the cells and make sure that they are developing in the right direction is extremely important eventually on a large scales of using cell therapy. And finally, this is another test. So if you take our monocytes and dendritic cells, do you have changes? Can you improve the flask versus the bags? So first of all, these are different markers of dendritic cell, activated dendritic cells, CD80, CD86 are very important and positive actor in the presentations of antigen to T cells. So in most of them, in all of them in this experiment, what you see is that there's not really change. So in a regular bag that they have made, that there's no deleterious effect of growing cells in bags. So now you can start using all the other factors I just presented to you, and then start building up something that would be much, much better. And this is what the team is doing now, and trying to still validate the matrices and how we can, with the bag now, with changing the structure of the physical structure, have a system that eventually will be closed and can be used in any hospital that they have a hood and a tissue culture hood and an incubator, and that a nurse could take the end of it and then take the cells and inject into patients. Obviously, this will not touch, these types of idea will not really work with the CAR-T, where you have to genetically modify cells, where you have to, select for a subpopulation of it. This is something that would be very different. But what the technology of the dendritic cells we're talking about, it's very much a nice directions and a proof of concept that all of these components may be helpful for some immunotherapy. And finally, I forgot that slide, by the way. So, but what you see is there's different combinations, and they look at different cell type. So they're looking at all of this, how they grow, how they differentiate with different matrices. So fluoropolymer in the bag versus so the cells which are moving in it versus a tissue culture flask. So all of these components of the work is tedious, but has to be done. And I think they have everything they need in the lab of Korean and the partnership with Saint-Gobain to do that. So I gave you an example of fundamental research that is done at McGill, and apply engineering and preclinical work. And eventually, we're hoping to start this clinical trial. We were hoping to do that in January, but obviously the COVID will delay things. But we are ready to do that, and we know exactly how which patients and how we will do this forward, and we hope that this will work forward. So I want to thank you all. Especially Vanessa, Craig. I didn't put your name there because I just talked to Vanessa, Craig.
That's okay. You showed my picture in that last slide.
I show your picture. Oh, that's true.
I'm the bottom right,
That's
extremely
you.
professional
Which one?
gentleman there.
Which one?
Bottom
On this
right.
side?
That's Stephen Marco on the left, and that's me on the right.
On the right here.
Yeah. So
Okay. Oh,
apologies,
you have it.
everyone. I guess you keep seeing me over and over and
Yeah, you
hearing my
know,
voice.
it's your beard as gray. This is.
Well, yeah, that's it. A mask does wonders for appearance, right?
So I want also to thank the OSLI and my lab to work on this. Nathalie Efekete, who has been a fantastic partner at Saint-Gobain to make sure it moves forward. Linda Pelletier and Pierre Laneuville for the cell transfer lab. The entire executive members of the McGill Regenerative Medicine, of course, Marine, that we cannot let her escape, and Daniel Boutillier and Mathieu Blanchet, two people at the at Canier that are developing this technology. Thank you very much. Hopefully we have a bit of time to have questions.
Thank you so much, Michelle. That was an outstanding presentation. The wonderful coverage of many different topics kind of blended together. And we really do appreciate the case study as well, the example of where you've come or where you've taken everything. I can honestly say a sincere congratulations, especially to you and Marianne and to everyone else involved. It's so somewhat humbling to see how far this has come from the initial concepts that we talked about earlier on in your presentation. And you've all done a wonderful job of this. So I'm very pleased to say we've got some questions coming in on the chat. So before I get into
Do you want to stop the presentation, to stop sharing?
well, it's fine right
Maybe
there. Maybe
we'll see.
some people seem to have some questions on DCs.
Okay.
Maybe it'll help guide the discussion. So,
Okay.
Anne, thank you very much for your question. Very nice presentation, Dr. Trombley. How many dendritic cells will a patient receive, and will one fusion be enough?
So the one infusion will not be enough. I think most dendritic cell immunotherapy have gone to 3 to 5 injections in patients. So this is quite well known. It's quite well known because there were several companies doing this. Provenge is still one of the immunotherapy and dendritic cell immunotherapy that's still going on. A very poor one, I must say, and it's not very exciting for oncologists for different reasons. And I could easily defend the technology we have. As far as the number of cells, I must say that this is something we look. I'm not the one who did that so much, but in the protocol we have a certain number of cells. If you I don't want to give you a number and I'm wrong, but if you're interested, we follow very much again the other dendritic cell vaccine that people have used. But it's enough. We calculated from the monocyte that we will have enough monocyte in 4 10 ml tube of blood to get at least 3 to 4 of transfer. So and the technique of maturing the dendritic cells are extremely efficient. It's not the one that people you see have used in other technology. MPLA is a great component in this, plus the inhibitor of the phosphatase. So that helps in reducing the number of cells needed.
Maybe if I can piggyback onto that question. So is this part of a clinical indication-based decision process on how many more infusions to perform? Or is it part of like the initial protocol?
So I think this would be more in the second phase. In the first phase, to make sure that we will fit in what was published already and used, we follow the guidelines of other clinical trials approved at NIH and Health Canada. So we have mainly followed that up. The biggest difference was that we don't need so many, because at the end the number of cells differentiated and mature are slightly higher with the compound in this protocol. So the number of cells you have allowed ready to go is the same one that people have used, but with using leukapheresis. I have a problem with this thing in my life. It's like my son with other things. But the point is that we don't need necessarily to have this system and have the donor or the patients coming for a long period of time. We can use just blood tests, so a blood tube.
Very good. Patrick has a question about the characterization. So what type of characterization do you expect to need to release each lot? As in, are you hoping to rely on the processing validation or assessing each lot?
So we have on the phase one, that's important. There are things that needs to be done by Health Canada. For example, the sterility is still there. So many, some of you will know that very carefully. 8 days is not so long for sterility test. And so there's issue there that perhaps Health Canada has raised, but I think we may have to go back to that. As far as testing, we also can take cells and look at the productions of IL-10, IL-6, IL-12. IL-12 is very important for the activity of the dendritic cells. IL-10, however, when they maturate, if the dendritic cells, mature dendritic cells, are producing a lot of IL-10, it's because they're not totally mature. And this is one of the issue of the Provenge vaccine. So we were very happy with our preclinical work and what we've done so far to see, and we did give that in the product in the document to Health Canada that the cytokines that we've tested are very clean, and it shows that the cells are healthy. So I'm not sure I can tell you more about that, and that's what it is.
That's fantastic. We have a couple of questions here. If we take a step back from the actual the case study that you're talking about here with your dendritic cell therapy, but one that focuses around the regenerative medicine program at McGill. Somebody's asked about admission in case there's no practical experience. So someone with a strong academic background in biotechnology and worked with some biomaterials production, but are there any specific requirements for entrance into the program?
No, if you are accepted in a graduate program at the Faculty of Medicine and engineering, you know if you have the grade to come in, and you should be able to take this program. Remember that there are specific courses that part of other departments in physiology and also so these are credit that you have to take. The only the two new course on regenerative medicine that you take, there's I think 4 credit. That's the 2 that you have to take to get really the stamp on your diploma. So what we're hoping is that they will learn a bit about that and be able to go to other places and join the larger community that is building around the world.
Excellent. Well, in your presentation, you had an excellent point about synergies between training programs and the foundational knowledge that's being delivered. And, it's vital that everyone understands as well that, we can't deliver the type of content that CellCan's focused on here without that appropriate training and foundational background from groups like yours and this regenerative medicine program at McGill. Ultimately, it's about qualifications, right? And so that's something that's very important that people understand. You do need a specific degree requirement to work in the field. If you're keen on getting into cell and gene therapy manufacturing, it's vital that you have the relevant foundational minimum qualifications, we call them, in order to take on additional role and training. So what we've done, it's very complementary to the program that you're putting forth at McGill, adding in components around the core foundational knowledge of stem cell biology, various projects, cell therapy projects, and in promoting them towards the clinic and getting them into patients, and I'm sure even some clinical trial examples, and as well the process engineering, which is critical around all of this. You hear so many from the regulatory perspective say, the process is the product, it's very true. It means everything that goes into it is what your final product is, especially for those that are regulated as drugs under our regulations and in other jurisdictions. And what we've put together are more specific skills for what you would need to work within the GMP environment and in support of that, whether it's quality team, executive, or even an operator as well. So I think the complement is fantastic.
Yeah. It's very important to have hands-on, and I think for producing these cells and your training, people have to look at the cells. They have to, it's not only facts. Experience on hands-on is very important. So
absolutely,
what you're doing.
It's all about, you spend a lot of time in the classroom training HQP students on how to become
Yeah,
scientists. And, it's very much the same. Once we go to the next level and you need to get into manufacturing, we have to train you on the job specific roles. It's actually a
absolutely.
regulatory requirement and it's a
Mhm.
continual need throughout your career, but as well helping to, get that career progression and stack your skills upon them. So I'm aware we've come to the end of our time. Thank you everyone so much for joining. If you can please take the time to complete the survey, the link is provided here in front of you. You can scan the QR code. We'd really appreciate that. Thank you so much, Michelle. We send our best to Corinne and family as well, and we thank her
Thank
for her
you.
contribution.
I'll pass
And
the message,
thank you.
and I really enjoyed coming. Yeah,
We look forward
thank
to
you very
talking to
much.
you more. Take care,
And thank
everyone.
you for everyone. Yeah,
Merci beaucoup. Thank you all.
merci
Merci
beaucoup.
tout le monde.
Close this.